Okay, welcome back everyone to the Immunisation Coalition's 2021 annual scientific meeting. I'm the chair for the next two sessions. My name's Margie Danchin and I've taken over from Lou Irving. Just a little bit of background about me. I'm a paediatrician and clinician scientist at the Royal Children's Hospital and Murdoch Children's Research Institute and University of Melbourne. And I'm the leader of the vaccine uptake group and have over about 15 years experience in immunisation related research. So we have another fantastic session for all of you this morning, session 4. The three speakers will speak one after the other, and then again we'll have about 20 minutes or so for questions. All right, so without further ado, let's introduce our first speaker for today, and that's Associate Professor Michael Murray. Professor Murray is the Divisional Medical Director of Continued Care and Head of Geriatric Medicine at Austin Health, and he's recently been appointed as the Geriatrician Lead to the Victorian Aged Care Response Centre for COVID-19, and he's going to speak to us about aged care, the weakest link. Welcome, Michael, over to you.
Okay, look, thanks very much for the opportunity. I'm most appreciative. I'm a geriatrician who is involved with BARC and also on the Aged Care Advisory Committee for the HPPC. The question topic was really aged care, the weakest link. And certainly there's no doubt that aged care was at most risk for lots of obvious reasons. But as I'll argue, part of the issue is how we got there. People would be aware that we entered the pandemic in terms of aged care already in trouble. People have called it a perfect storm. In fact, it's not a perfect storm. We really just lit the fuse of 20 or more years of neglect. Residential care, is it a question of cognitive dissonance or hypocrisy? And it's probably more hypocrisy. At the end of the day, we've walked past the problem so often, or more often than a wooden horse on a merry-go-round. We've had about 17 investigations, reports, commissions, etc. And indeed, when I was appointed as the interim chief clinical advisor to the Aged Care Quality and Safety Commission, I was handed over a number of large folders of all of the previous studies. And it was clearly evident that there was no new issues. It was simply we weren't actually dealing with them. There are major issues with the health sector and substantial workforce issues. And this is not unique to aged care, but it was certainly much worse in aged care because of the pay scales and pay rates and because of the economic situation in residential aged care. There was a vulnerable care environment. It was surprising that there were still residential care facilities with 8 people in a single room. There's only one that I'm aware of, but there was certainly lots of 2s and 4s. And this has major IPC issues. This is a bit similar, if you might draw the analogy to subacute care. When I was a junior doctor, if you wanted to find administration in the new hospital you'd been rotated for, you used to look for the new carpet. If you wanted to find aged care, you looked for the oldest building, usually away from the new building. There were significant problems within the community, and as people are aware, there is lots of supply issues, and community places are currently got a backlog with a waiting list of in excess of 12 months, certainly for the higher packages. And this really set aged care up to fail. What happened after wave 1? Well, I think we were complacent. We didn't imagine, or perhaps we didn't imagine a bad enough scenario. We thought we'd prepared adequately, but in fact, much of it was desktop audits, telephone audits, etc. And we didn't listen to the harbingers of doom, and they were marginalised, and they've become marginalised. What happened in wave 2? Well, we set up VARC, Victorian Aged Care Response Centre. There was a substantial pandemic sector response, and public health nearly fell over. Are we prepared in the future of all jurisdictions? Well, not yet, but we're on the way in some areas. The sector nevertheless remains under severe stress, and although there's been some temporary funding, we haven't actually fixed the problem. So back to pre-COVID. People will have read or may have read the Royal Commission, the Council assisting its final submissions, and people are aware the final report's due on the 26th of March. It was entitled A Tale of Shame, and that pretty much should tell you all. It talked about ageism, it talked about distressing tales of poor care over thousands of submissions, and perhaps the most distressing was that almost all of the issues and problems that we identified had been previously identified in the many previous reviews. And this wasn't any particular persuasion. All governments had marginalised the issue, and surprising given that over 25% of people have a direct relative or will experience a direct relative in aged care, it's really quite amazing how we could ignore the problems. I'm fine, I expect to just drop dead. What was inadequate? Staffing. Clearly, staffing was a major problem, and it was inadequate training coupled with substantial turnover. And it's not uncommon to get somewhere between 18 and as high as 24% staff turnover in a single year. Training becomes hugely problematic in such a situation. Now, many of us who work in public hospitals will also be aware that prior to COVID, we had a substantial turnover in our nursing staff as people moved in and out of the sector into other areas, travelled overseas, etc. And that again provided challenges. Nevertheless, it was much more of a challenge in residential care for lots of reasons. And also the way we're actually undertaken the staffing, and increasingly because of economic pressures, as we'll talk about later with staff working across multiple facilities, some providers had gone to 4-hour sessions. So if there's 4-hour sessions, what you'll find is that in a casualised workforce, many people work across multiple residential care facilities. There was systemic ageism, and of course, this is not in the positive sense. And there was really lots of problems with capping of resources, as I've mentioned. We came to realise that staff was the predominant link in terms of infection, and somewhere between 80 and 84%, with 25% of those seeded from other facilities, either through staff or through close contacts of staff. And this became a particularly important issue because what you would see in VARC, as we had 2 or 3 outbreaks a day, and then we were increasingly anxious as residential care facilities became overwhelmed, is that you would have a ripple effect from the index case or the index facility, and you would get progressive spread to facilities around that. Why did that happen? Because if people worked in one facility, the other ones they tended to work were ones that were relatively adjacent. So you saw a major spread up north, Epping Gardens, Estia, Heidelberg, St Basil's. They all started to spread out like ripples. It moved then more towards the northwest. It then moved more to the west, and then you've got a progressive smattering of east and southern. But it became a major problem. And why did this happen? Well, because you had close contacts. But why did that happen? Is because people who worked in residential care, of which there was at least 49 different ethnicities, there were large households, and frequently multiple members of that household worked in the same or similar service type industries. And therefore, you and I know that the likelihood of getting staff infected were actually roughly proportional to the baseline rate in the place they lived and worked. We've talked about staff, the casualisation of staff, because in the end of the day, if you don't work and you're casual, you don't get paid. So lots of staff worked across multiple facilities until we brought in some better controls and better support of casualised staff. It was very difficult for people not to work across multiple facilities in order to make an adequate pay for people on the base rate. They also tended to work surprisingly long hours in lots of facilities. And interestingly, larger facilities tended to have longer outbreaks, and longer outbreaks often had more links when we plotted out the links from staff to other facilities. So I stopped counting at 182 plus outbreaks because the tail end of the second wave was very different from the start. And if you watched the wave as it unfolded, inevitably what would happen was that staff and patients or residents became positive at roughly the same rate, sometimes staff first, sometimes residents, depends on where they'd caught the infection from or their source, whether it be public health, hospitals, or whether it's from staff. And the effect of a positive or more than one positive staff member very rapidly translated into multiple positive residents, and you got a literal explosion. Later on, after PPE and IPC were put in place, there was a substantially different model, and the last several outbreaks, in fact, the last dozen or two outbreaks, were really one or two staff members, occasionally a contractor, and they almost never were associated with a positive staff member. And it just showed the value of IPC and PPE. The majority of cases, nevertheless, were really only one or two cases, but of major concern were the cases that had multiple contacts and multiple infections. We plotted out the activity and what we did when myself and Professor Mary O'Reilly joined the prevention team in VARC in late July. The first thing we did was to negotiate the introduction of PPE. Now that was a game changer in reality, the combination of PPE and ADF prevention visits, of which we're set up, because there are a number of things seriously going pear-shaped. And this is just an example of how the ripple effect and how things spread across the three hubs and how we tracked it. There was initial mixed messaging, and it was very much hospital focused. For those who work in hospitals, you would have got a daily or twice daily update of exactly how much PPE you had. And we had knowledge of the sizes of gloves, the type of gloves, and how long the gloves would last with current utilisation. And this was a major source of concern and watching. The information in the sector, the residential aged care sector, was exactly this. PPE is a limited resource, so please only use when it's needed. And the specific link in the advice was that when you are symptomatic. And of course, that's a disaster, because by the time you're symptomatic, as you and I know, you've been infectious for probably 24 to 48 hours. And we know from experience that when we send out sort of SWAT teams to residential care facilities where a single staff member had become, a patient was found to be positive, just on random screening in order to move facilities, we found that several residents were already positive. In effect, this actually made the concept or the idea that you would pull out the index or NIDAS case fraught. And quite clearly, in the absence of PPE and IPC, it simply wouldn't work. And there's a number of similar cases, and I think there's an article in currently awaiting publication to really go through that in some more detail. But that's basically what we found. And there was variable interjurisdictional advice, is probably putting it mildly, because there was a huge issue with harmonisation. And I think there were at least 5 different mobile teams, all of which who had a slightly different take on PPE and IPC. And residential aged care, vulnerable at the best of times, became very confused. We had, as I said, a significant problem with supply. We had lots of issues with recommended product, N95 versus KN95. We had, let alone the issue of fit checking and fit testing, which really came very late in the day. And the reusable versus single use. Obviously, if you're on overseas chat groups, you'll be aware how difficult and distressing this is. And people talk about carrying their own goggles, etc., even their own masks in a plastic bag and reusing it over several days because there's insufficient supply. So we got slow harmonisation, and we had a challenge figuring out who was actually responsible. And it took several days to progressively organise this. Whilst this was happening, obviously residential care was going from bad to worse. So what did we learn? Really major issues of workforce. And what would happen almost inevitably within 36 hours, and usually often by 24 hours, that a substantial number of staff would call in sick or would simply not turn up for work. So one facility we went to very early on, it was pretty typical. There was only 2 care staff that turned up out of 7 on day 1 after the infection was announced. And this became increasingly challenging and problematic as public health basically put staff into quarantine. And we had a huge problem with getting staff. So staff failure became the major issue, and care failure, not illness, was the major reason early on why people got admitted to care. We also had a major problem with leadership at a facility level. And the reason this was a major problem was because at the end of the day, if the leaders of the organisation were unable to work, they were sick or furloughed, then it became a major problem because there was nobody who actually understood the process. And it became hugely challenging and problematic to actually insert leadership. But if you didn't, you couldn't actually keep running the facility. And logistics became a major issue, and people are well aware of the plastic, the yellow bag issue in Victoria, where this became a major political football and a major cause of distress, where a huge number of plastic bags accumulated, and it caused a big challenge for our politicians, but also a big practical challenge of how you're actually dealing with this until people came up with the concept of getting containers, putting them in containers, and then shipping them out to get burnt or incinerated. It was a real challenge around speed and adequacy. As I've said, responsibility was a major problem, and people would be aware that responsibility for the care facility rests with the proprietor. There's a complex system through Aged Care Quality and Safety Commission and through public health around appointing under a notice to agree additional clinical support, or through public health actually bringing public hospitals and the resources into play. This took a little while to sort out, and it took a little while to negotiate, but it often became an amazingly effective tool, often well received, but not universally, by the facility themselves. Rights, choices, and engagement was a really big problem. We didn't fully realise it. I'd be embarrassed to say at the start, whilst we're desperately scrabbling to try and fix the system. Residents and information, family information was hugely problematic. The advice to facilities is you've got to expect up to 1,000 calls a week. And of course, if you've got reduced staff numbers and you've got residents who are unwell, and you've got to get into and out of PPE, and therefore you need about 1.5 times the staff you had pre-COVID, then you can rapidly guess that you don't have anybody or inadequate people to man the phones until dedicated people are brought in to do that. General practice was a huge challenge, and it remains a challenge going forward. I've called it a tale of two cities, and you'll remember the opening line for A Tale of Two Cities: the best of times, the worst of times. One of the centerpieces of public hospital response, and indeed GP response, and I personally have found this hugely beneficial in dealing with my specialists, is the use of telehealth and telecommunication. In effect, my belief is that during the crisis and during the pandemic, it was largely a failure. Why was it a failure? Is because you had nobody to hold the camera. You had no staff to go around and actually act as the local clinician in order to support the GP's intervention. So the absence of face-to-face became hugely problematic. You also had the problem that many of the GPs in the traditional model, many of which were older themselves, and many of which were unable to or unwilling to attend the facility. And therefore, they're really only left with telehealth. There's no doubt that telehealth was hugely beneficial as a communication tool for case conferencing and handover and preparation, particularly in conjunction with in-reach type services, in-reach outreach, called various names in various jurisdictions, but basically health clinicians coming from usually the local public health sector. Nevertheless, whilst there were some stunningly good examples of GPs who virtually led the care of the residents in particular facilities, for many, particularly where there are a large number of GPs involved in any one facility, this became a significant problem. And the many GP, few patient model was a major challenge. What contributed? Well, as I said before, we didn't actually imagine. You might say, well, why didn't you imagine? Weren't you looking at the Italian experience at the time, which was clearly evident? Yes, we were, but we really thought that Australia as an island was well protected. We really thought this wouldn't happen to us. It sounds like hubris or arrogance or foolishness, but that's what the truth was. And we thought we were well prepared. We had done lots of desktop audits. We had sent out lots of advice. We thought, whilst there was clearly some risk, we didn't fully appreciate how rapidly this could spread. We inherited a model unable to cope and already not coping in financial crisis with a large number of facilities, basically even excluding depreciation, going backwards. We had a poor environment in many facilities. We had inadequate staff skilling and inadequate staff access. And finding extra staff became a huge issue, and Victoria became became the beneficiary of support from Western Australia, particularly Queensland, from Bolton Clark, from a large number of providers, and particularly from private hospitals as they cut back their elective surgery and they were able to support a residential care. They're also able to admit some patients or some residents who became patients, usually not through clinical necessity, although sometimes, but through failure of care. And they really did a huge job and stopped public health, which was only sometimes days away from falling over. They stopped it falling over. We had really problems with poor staff engagement, and we had problems with poor staff engagement at lots of levels. And I think until you've actually done lots of Zoom meetings with staff who have been COVID positive and who are at home distressed, you don't fully appreciate the interdependency and the support that people get from their supervisors. And when these people are also lost, how discombobulated they effectively became. We also got a huge amount of negativity, and people will recall the issue of hospital heroes and facility villains. My public health service got no end of gifts. Residential care got no end of criticism. Facility size makes a difference. Pick a small one. Residential aged care has been under stress. The in-reach models of geriatric care was not set up to succeed. Large aged care groups, not only large facilities, didn't necessarily do better. GPs had a problem, and you needed a controller to act decisively. In-hospital care needs to be reviewed in light of outcomes. The only thing not in short supply was distress, anger, and blame, and you burn your friends in a pandemic.
Thanks very much, Michael. You've certainly raised a huge number of issues there, and we've got some good questions in the Q&A function, which we'll come back to and really, I suppose, try and look at some of the learnings moving forward. But I'd like to introduce our next speaker, Professor Katherine Kedzierska. Katherine is an immunologist and head of Human T-cell Laboratory in the Department of Microbiology and Immunology at the Doherty. And Katherine's going to talk to us now about immunity to COVID-19. So thanks, Katherine.
Thank you, Margie. And I'd like to also thank the organizers for the invitation. I'm really delighted to join the meeting today and share with you some of our findings on immunity towards SARS-CoV-2 infection. So our journey into COVID research started on Saturday, the 1st of February, when we obtained blood from one of Australia's first COVID-19 patients. We've done analysis on longitudinal sampling in this patient, and we're really fortunate on the 16th of March to publish the world's first case report of immune responses in non-severe COVID-19. After that, we sampled COVID-19 patients through the first wave and the second wave of the pandemic, and these are the results I'm going to share with you today. So in our, change my pointer. So our first patient was a previously healthy female, 47-year-old, who developed symptoms 7 days after arriving from Wuhan, China. On day 4 after disease symptoms onset, the patient self-presented herself to the hospital. Viral RNA was detected on day 4, 5, and 6 after disease onset. The patient recovered on day 10 and was discharged from the hospital on day 11. In the lab, we've received blood samples at day 7, 8, 9, and 20 after disease onset. And we received the patient blood via the CETREP platform led by Irani Tevarajan. So first of all, we decided to look at antibody secreting cells, which are short-term effectors that are known to produce large amount of antibodies, as well as T follicular helper cells that help B cells to facilitate production of long-lived antibodies. And we stain for markers associated with those two cell types on day 7, 8, 9, and 20 after disease onset. And here one dot represents a cell. So if you look at the gate, within those gates on day 7 after disease onset, we found small but distinct populations of both antibody secreting cells and follicular T helper cells. However, really exciting findings were on day 8, when we could see really large populations of both of those two cell types. And that was exciting because we knew from our experience when we studied immunity in patients hospitalized with influenza, that such increase in those two populations usually meant that the patient would recover within the next 3 days. And that's what happened in this case. We further look at the activated killer CD8 T cells and helper CD4 T cells. And again, you can see on day 8, we found a marked increase in those populations. And looking at the cytotoxic killing capacity of those cells, especially CD8 T cells, we found large proportion of those cells displaying cytotoxic molecules, suggesting that those cells were elicited into the immune response and ready to kill virally infected targets. So really, the patient had a textbook example of immune responses to viral infections. And those immune responses really nicely controlled inflammation, that as in this patient with mild to moderate disease outcome, we found no dysregulated cytokine chemokine profiles in the blood or no inflammation. So following our first case report, we obviously wanted to decipher immune responses at a high level of resolution in a large cohort of patients. So we really asked 2 questions. One, what are the immune responses preceding patient's recovery? And the second one was, what are immune perturbations associated with severe disease? And these are my amazing COVID-19 team who did lots of work in the laboratory. So we've recruited patients via the CETREP platform with Irani Tevarajan from our own DZ cohort at the Alfred Hospital in collaboration with Alan Cheng and Tom Kotsimbos, as well as at the, in the second wave via the drastic cohort at the Austin Hospital in collaboration with Jason Chabiano and Claire Gordon. So in our analysis, I'm going to be showing you 74 samples from 26 hospitalized patients. They include longitudinal samples, ICU, ward patients. We also had community COVID-19 patients from 50 individuals and healthy donors, 20 to 49, depending on the panel. So firstly, we verified activation of those 2 important T cells, important subsets, antibody secreting cells and T follicular helper cells in a large cohort of COVID patients. So in comparing to both healthy in gray or convalescent COVID patients, you can see that acute disease, we see increased, statistically significant increase in both proportion and numbers of antibody secreting cells and T follicular helper cells in patients' blood. But now, because we had a large cohort of patients, we were able really to define the kinetics of those cell activation of those cells. So in pink here, you can see that the peak of antibody secreting cells is around day 9, and it's a sharp peak followed by a decline. In contrast, the peak of follicular T helper cells occurs 2 to 3 days later, and it's broader. And it's not to say that COVID is the same as influenza, as they are obviously two different diseases. But looking at the kinetics of those immune responses, they both very similar. We further verified activation of a broad array of T cells, CD8, CD4, and CD8 T cells by those two markers, CD38 and HLA-DR. Looking again at the killing capacity of those T cells to kill virally infected targets by assessing cytotoxic molecules, granzymes, and perforin, you can see that especially within the killer CD8 T cells, the activated type, the majority of those cells were armed in the cytotoxic molecules. As you may remember, in our first case report, we found no dysregulated inflammation, cytokine, chemokine production in the patient. However, here now, when we put together a large cohort of patients, we did find that at the acute COVID-19, there is an increased level of IL-6, IL-10, and IL-18, which was also reported by several different groups. So when we put together the 184 immune features that we analyzed in our study, you can ask what are the main key features that are different between acute and convalescent COVID-19. You can see that it is really activation of a broad range of immune cells that really drives recovery from COVID-19. Those cells are not activated at short-term convalescence, and we usually look around day 30. But here we started seeing memory B cells and memory T cells appearing. So apart from cellular immunity, obviously antibodies are really important. And here very early in the pandemic, Florian Kramer from Mount Sinai in New York, he pioneered RBD ELISA to detect antibodies in patients' plasma, and Brendan Chua in my lab established that assay. So we incubate, pre-coat the plate with RBD, and then incubate patients with patient plasma. And more color we see, more antibodies the patients have. And when we analyze our cohort of COVID patients, what we found the seroconversion rate higher than 98% for IgG at convalescence. IgM and IgA were around 60%. In collaboration with Kanta Subero, we also look at the neutralizing activity of those antibodies and found that a substantial proportion of COVID-19 patients, the antibodies had neutralizing activity. And you're seeing that here in gray and black. However, interesting results were when we ranked the patients with regards to the neutralizing immunity. And what we've observed was that patients with the highest neutralizing activity had all three types, IgG, IgM, and IgA of antibodies, possibly suggesting that engaging a broad range of antibody isotypes can somehow lead to increased neutralizing activity. So we know that in our cohort, patients had a range of antibodies. So the next question we asked was, during the acute time point, during hospitalization, is there a correlate? Can we find a correlate that would then predict antibody responses at convalescence? And the answer was yes, and there were circulating T follicular helper cells type 1. So what I'm showing you here are statistically significant correlations between the activated follicular T helper cells type 1. And you can see they really strongly correlate, and that acute time point really strongly correlate with spike IgA, IgM, and IgG, as well as the neutralizing activity from the of the antibodies, and less strongly with the RBD antibodies. You can see it here also shown by a different, by a bubble plot. You can see really strong correlations between those follicular T helper cells at acute time points and spike antibodies and neutralizing activity. So we know that TFAGs can correlate with antibody responses. What about the avidity? What about the quality of the responses? So we assess the quality by looking at avidity of the antibodies. So here, when we compare between sampling, so usually that's on hospital admission and hospital discharge, you can see that the avidity, the quality of antibodies increases for both IgG and IgM. And when we look compared cellular events with the quality of antibodies, we observe again statistically significant correlations between the follicular T helper cells type 1 and RBD IgM. So we know that follicular T helper cells are really important in predicting antibody responses. What about the classical CD4 helper T cells and CD8, the killer T cell responses? So to understand that, we incubated cells from the patient's blood with peptides spanning SARS-CoV-2 membrane, nucleocapsid, and spike. And here we're looking at really activated CD4 T cells towards those SARS-CoV-2 peptides. And what we've noticed is that all the patients, the vast majority of patients had CD4 all T cells. However, CD4 T cells dominated over CD8 T cells. And to further understand those weak responses by CD8 T cells, we identified some specificities, peptides. Here we're showing you one associated with the dominant HLA-0201 phenotype. And what we found, the results were quite disappointing, because looking at the responses of those CD8 T cells in acute and convalescence, you can see that they really didn't differ that much to those found in healthy, of naive pools found in healthy. There was only around 5-fold increase above what we've seen in healthy pre-pandemic controls. And those at convalescence towards COVID-19 were much lower than the memory pools in both in healthy individuals towards both influenza and EBV. And that really suggests suboptimal SARS-CoV-2 specific CD8 T cell response. However, it's not all bad news for CD8 T cells, as we and others have identified a really prominent specificity associated with HLA-B0702. So when it comes to SARS-CoV-2 and CD8 T cells, it is good to be HLA-B0702. So we know that during COVID, in patients with mild to moderate infection, there is a broad range of robust immune responses that drive recovery. But what about immune perturbations during severe COVID-19? So to define that, we compared immune responses in patients that were admitted to the ward versus those that were admitted to ICU. And we're looking here at activation of different arms of immune responses. And you can see really increased frequencies of activated T cells, NK cells, monocytes in ICU patients. And if you look here, for example, on CD4 T cells, what it shows you that some patients in their blood, close to 90% of all the CD4 T cells were activated, meaning that there is a broad overactivation of immune responses in ICU patients. We also found increased levels of IL-6, IL-18, and soluble IL-6 receptor in ICU patients comparing to ward. But striking observations were that soluble IL-6 receptor level were not associated with increased IL-6 level in either ward or ICU patients. So there were some patients with high IL-6 but low soluble IL-6 receptor and vice versa. And obviously that's got really important implication for the anti-IL-6 receptor therapy. And we suggest that both IL-6 and soluble IL-6 receptor should be assessed in patients prior to the therapy. So similarly to how we compared immune responses between acute to convalescent patients, now we plotted a volcano graph to find the key features that were different between the ward and ICU COVID patients. And again, you can see that the main features that were different were this really highly activated gamma delta T cells, CD8 T cells, CD4 T cells, and other immune responses in ICU patients, again suggesting that in those patients the immune response was totally on overdrive to the point that was most likely detrimental. So again, we've plotted all of our immune features of the cell types against the cytokines that we found in individual patients. And we found that IL-6, soluble IL-6 receptor, IL-18 hypercytokinemia was associated with dysregulation of both innate and adaptive immunity. And here are some of our correlations. So in summary, what we defined immune responses in mild and moderate COVID-19. So what we found that those patients have increased levels of IL-6, IL-18, MCP1, interferon gamma. However, this is quite a regulated cytokine response. Patients had really prototypical antiviral immune responses that we have observed in other viral infections like influenza and others. We've seen high but really transient activation level of monocytes, NK cells, gamma delta, CD4, CD8 T cells. We've seen antibody production, engagement of follicular T helper cells, antibody secreting cells. And an important finding of our study was that activation of T follicular helper cells type 1 at the acute time point really nicely correlated and could predict antibody responses at convalescence. In contrast, in severe COVID-19 patients that were in ICU, we found really dysregulated cytokine response, and they were mainly IL-6, IL-18, and we found elevated level of soluble IL-6 receptor. And that led to immune hyperactivation, really prolonged and high level activation of immune cells, suggesting again that the immune responses in those patients were really on overdrive. So I'd just like to acknowledge everyone in my lab who worked really hard last year, but we've been really privileged to be part of a global effort to understand immune responses in COVID-19. We've had numerous collaborators within the Dorothy Institute, University of Melbourne, work closely with CETREP ID, WHO, Kenta Subarao, Alfred Hospital, Austin Hospital, and like to acknowledge our funding body. Thank you very much for your attention.
Thanks so much, Katherine, for that extremely elegant presentation. We'll come back to some questions after Dave's talk. Next, I'd like to introduce Professor David Burgner, who's a paediatric infectious diseases physician at the Royal Children's Hospital in Melbourne and Murdoch Children's Research Institute and University of Melbourne. And he's been at the forefront of our research response, COVID research response on our campus. So Dave's going to talk to us about COVID-19 in children, curiouser and curiouser. Thanks, Dave.
Thank you very much. Thanks, Margie, and thanks very much to the organisers for giving me the opportunity to join such a stellar cast. When I got the invitation to a conference, I kind of went back into pre-pandemic mode and thought that, you know, there'd be some foreign travel, perhaps, I'll be sipping wine on the flight, and then I realize that it's time to get real, and we're dealing in pandemic times. What I'd like to talk about today is a bit of a change of tack in terms of looking at a couple of aspects of COVID-19 in children. Firstly, to look at whether and why COVID-19 is less severe in children, and then to talk about PIMS-TS, this new syndrome that caused a lot of excitement with us here within pediatrics. So as you know, we've had an epidemic not only of the virus, but also of publications. Nigel Curtis tells me there's a COVID-19 paper every 5 minutes last year, and even making sense of these huge number of papers takes on a slightly surreal turn at times. But it became clear quite early in the pandemic that children were less infected than adults, and less severely infected. And the question was why? And that's an important question, because it obviously may give us some insights into how we might understand the disease in adults and those at higher risk, and how we might prevent and treat severe COVID-19. So lots of studies, lots of small case reports, but pulled together into some neat meta-analysis. This is one from JAMA Pediatrics, showing that in contact tracing studies, children, and to a lesser extent adolescents, were less likely to be infected with SARS-CoV-2 and less likely to seroconvert. Obviously, that may reflect to some extent the performance of serology, particularly in children, which I think is outside of my remit, but is an interesting issue as we move forward. So there have been relatively few big case series of children admitted to hospital with COVID-19. This was a series from the UK of 650 children, and it was difficult to know because this was relatively early in the piece, and children were admitted for various reasons. And I suspect at times children with comorbidities were admitted even if the COVID-19 was not a driving reason for admission. But what you can see here is that about 1 in 5 were admitted to PCU, very low mortality rate, and all the children who died had a profound comorbidity. So it didn't sound like from this report that they were dying from COVID-19. And if you look at the symptom distribution in pink, you can see that about 2/3 of the children had fever, but then the other symptoms of these admitted children were actually increasingly rare as you went along the different types of symptoms. So really, largely apart from fever, and this is an admitted population, an asymptomatic infection. They did a neat heat map of the different types of symptoms, and some clusters emerged. So the most common cluster was a respiratory cluster, perhaps not surprisingly, in children, though a pretty mild one for most of them. And then this what they called a systemic mucocutaneous enteric cluster, which, as you will see, resembles this new hyperinflammatory syndrome and looks like it probably is the mild end of the spectrum. And then this neurological cluster, which is particularly rare. Now, more locally, we got interested in very early in the pandemic about this idea that children were different. This is the most amazing family. They're incredibly photogenic, which helps, but they were very interesting. So the parents had gone to a wedding interstate, early in the pandemic, become infected, became mildly symptomatic, they weren't admitted, came back, and their 3 children were heavily exposed. The youngest basically shared a bed with the parents when they were symptomatic, but the children always swabbed negative, and we named them the Discorvid family. And it was really quite an interesting journey in terms of trying to understand what was happening with this family. And it was a great opportunity for collaboration, both within our institute, but also particularly with PDI, with the Doherty Institute. This was an amazing family. They had blood, every child and the parents had serial blood tests, and they actually had 11 nasopharyngeal swabs each during the course of this illness. And this was work led clinically by Shidan Tosif and in the lab by Mel Neeland. And they, in the lab, there was flow cytometry, looking at plasma and saliva antibodies, and then in collaboration with PDI, some systems serology. So as I said, both parents are symptomatic and seroconverted 2 children did have mild symptoms, very mild symptoms, but in fact, the 2 older children remained seronegative. And the 1 child who'd been sharing the bed with the parents never seroconverted. Did seroconvert. So no real correlation between symptoms and seroconversion. What was interesting though, work from Phil Sutton's lab at MCRI, that all the children and the adults did produce salivary IgA, which coincided with clearance, at least in those that were symptomatic, of their symptoms. The cellular immune profiles and the cytokine responses were almost identical in the children and the parents. And this family had quite important implications, we think, because it seemed that these children could be infected without virological and largely without serological evidence of infection. But clearly they had been infected and they managed to clear the infection. So it's a very interesting family, probably has implications for how we diagnose and trace children who are exposed to this virus. Now, so we know that SARS-CoV-2 causes less infection and is less severe in children than adults. Similar patterns were seen with CoV-1 and MERS, but this is obviously extremely different from almost all other respiratory viruses, where the burden does fall largely on children. And then why might this be so? And again, the literature is hard to sort out. Luckily, Petra Zimmerman and Nigel Curtis from MCRI, Petra's now back in Switzerland, have done a very nice review at the end of last year looking at this, and they divided this up into factors that increase severity in adults and factors that seem to protect children. And I'm not going to go through this in detail, but I do commend this review to you. So in terms of factors that make the disease more severe in adults compared to children, we know that although this is a respiratory virus, it probably hasn't read the textbook in terms of what a respiratory virus should do. And in fact, the pathogenesis occurs in the blood vessels and the endothelium, particularly with endothelial dysfunction and hypercoagulopathy. And those changes and endothelial changes are much more prominent as you get older, and they're much more prominent in comorbidities that are common as you get older, like heart disease, diabetes, etc. The other well-reported difference between adults and children is that both ACE2 receptor for the virus, for the spike protein, and cofactors are less expressed in children compared to adults. And there are some other interesting possibilities that are being raised, including the role of CMV in immune senescence, which may make adults more prone. In terms of factors that are suggested to protect children, little bit more unclear. Possibly the predominance, especially in young children, of innate immune responses of over adaptive, although that potentially could give you more inflammation. Cross protection from circulating run-of-the-mill coronaviruses might be important. I'm not convinced, but that microbiota will necessarily play a huge role in this, but I think that's a bit remains to be seen, and those are associated data. They're a bit difficult to interpret. A very interesting idea that bats, who obviously are a reservoir for this illness and other viruses, have very high levels of melatonin, and that might be protective. I've been religiously taking my melatonin every night as a result. So far, just to summarize, infection is clearly less common in children and less severe. The standard diagnostic tests may not identify all children are infected, and this fascinating idea that children may be infected without becoming PCR or positive or seroconverting. And there are many known unknowns, including long-term effects, and whether even this asymptomatic infection in children may lead to some version of long COVID. But overall, early in the pandemic, we were, as pediatricians, feeling reasonably relaxed. We might have dodged a bullet. We could sit down and have a drink. And then in amongst this ongoing deluge of publications, this alert came out of the UK in April of this new syndrome that was hitting their ICUs. This went out through the PICS Society in the UK, spread out to all GPs, and caused a huge amount of anxiety around the UK, and then was obviously picked up by the press very quickly. And it seemed initially that this might be related to Kawasaki disease, which was a particular interest of mine over the last 15 or 20 years, and of many pediatricians. It is a very fascinating disease. And my two worlds suddenly collided with this new syndrome. So this new syndrome has been named by the British as PIMS-TS, Pediatric Inflammatory Multisystem Syndrome, temporally associated with SARS-CoV-2. Nothing like British to make things simple. And it was renamed by CDC as MIS-C, Multisystem Inflammatory Syndrome in Children. You choose which you prefer. I think I go for PIMS-TS. And so what I'd like to do for the remainder of this talk is just talk about where this fits into the landscape, not only Kawasaki disease, but also toxic shock syndrome. What is this new syndrome? What is its epidemiology? What does it look like immunologically? And what's it telling us not only about SARS-CoV-2 infection, but is it telling us something about the sort of curate's egg of pediatrics, which is Kawasaki disease? I have to tell you straight up that it's not actually Kawasaki disease, but just to give you a bit of context, I should tell you a little bit, or remind you, those of you that are not Kawasaki fanatics, what Kawasaki disease is, and show you the world's only Kawasaki disease joke. Professor Kawasaki described this condition in the 1960s and died in the middle of last year at the age of 95. He was a giant of pediatrics. He first described this condition in a small regional hospital outside Tokyo that clearly didn't have a medical illustration department, and couldn't actually report it until the professor in Tokyo died, because he didn't actually believe that it was a new condition. Initially, it looked like a new viral exanthem, but then, as I'll show you in a second, it started to pick off children because of the coronary artery damage. So this is a systemic vasculitis of muscular arteries throughout the body. Even after decades of research, there is no diagnostic test. We don't know the cause, although we think it's one or more infections. And suddenly we have a rash, so to speak, of Kawasaki disease reviews from people who, I tell you, never have published a thing on Kawasaki disease. So we make a clinical diagnosis of Kawasaki disease, high fever, and a number of the cardinal or diagnostic symptoms that reflect and signs that reflect the vasculitis, but it's a difficult diagnosis to make. So these children, these are all children who might be thought to have Kawasaki disease, but in fact, only one of them has Kawasaki disease. The others have a variety of other conditions, other infections, and including acrodynia, which is mercury poisoning. Here's a particularly severe case. Kawasaki disease is important because it is not just another viral exanthem. It does cause damage for reasons that are not at all clear to the coronary arteries, and in bad cases causes these giant aneurysms that you can see here. These can occlude, and there is a small but appreciable mortality associated with Kawasaki disease. Now, related but much better understood is toxic shock syndrome, initially described in relation to retained tampons, but also now with other infectious foci. We know what causes this. It's usually due to group A strep and Staph aureus. The very few, if any, reports that I can find of viruses causing this super antigen mediated disease. And it shares some features, as you can see from the pictures, of Kawasaki disease, but shock is a cardinal feature, as the name suggests, as is gastrointestinal symptoms and coagulopathy, which is important when we come back to talk about PIMS-TS. It's characterized by a massive inflammatory response. Now, the two conditions do go together. It's been known for some time that you can get Kawasaki disease and you can get features of toxic shock syndrome. We described some cases from Australia some time ago. To those of us in the field, it's known as Kawasaki or Kawasaki shock syndrome, and it's relatively rare. It occurs in older patients. Kawasaki disease is a disease of infancy and preschool children. This is a disease of older children, mid-childhood, and is characterized by myocarditis. So where does PIMS-TS fit into this landscape of Kawasaki disease and toxic shock syndrome? So we have Kawasaki disease. We see a spectrum of severity. As I've said, we see some overlap with toxic shock syndrome. Somewhere in the middle sits Kawasaki or Kawasaki shock syndrome. And I think this is where PIMS-TS sits in that sort of area. As I said before, it's thought to be distinct from Kawasaki disease, although it clearly shares some features. And I'll just show you some of the differences and similarities now. So when we talk about Kawasaki disease, there's most of the money and most of the research has been looking at which infection causes Kawasaki disease. And that's been the holy grail, if you like, of Kawasaki disease research, one of the great mysteries of pediatrics. And there's a long list of things that don't cause Kawasaki disease. And about 15 years ago, a then fashionable human coronavirus, coronavirus New Haven, appeared as a possible cause of Kawasaki disease. It caused quite a stir, published in good journals, and editorial from Ken McIntosh. And like every other proposed cause of Kawasaki disease, that was quickly disproven. So we still don't know what causes Kawasaki disease. One of the interesting questions, obviously, that came up almost immediately with this new syndrome was, not only is this the tip of the iceberg? And I have to tell you, it's quite hard making jokes when you can't hear anyone laugh. I guess I should have been used to it by now. But had we found the cause of Kawasaki disease, or had it found us, were we about to see a Kawasaki disease epidemic? And were we about to see a PIMS-TS epidemic, which was much more worrying because there were some children dying in the initial part of the PIMS-TS story. So overall, there's been no increase in Kawasaki disease incidence. In fact, there may have been something of a drop-off for reasons that we can go into. And interestingly, in the very high incidence countries for KD, Northeast Asia, other parts of Asia, there's been virtually no PIMS-TS cases reported. And the epidemiological, clinical, and immunological features between these two conditions, PIMS-TS and Kawasaki disease, suggest that they are actually different, distinct phenotypes. And I suspect they have downstream shared pathways that make them look similar. And these are data from PEDS, the surveillance network, who were already surveying actively for Kawasaki disease as part of ongoing research that we were involved in. And this is just to show that there's really been no tick up, if anything, a slight drop off in Kawasaki disease numbers since the COVID-19 pandemic in Australia. So the epidemiological features of this new syndrome, there were a number of case reports, and then a case series came out of London, which was particularly badly hit because obviously the first wave was particularly severe in London. And it appeared to occur predominantly in those of Black, British, African and South Asian origin, about 60%. Now, there may be many reasons for that, including overcrowding, relative disadvantage, access to healthcare, but that's a consistent finding. And clinical and laboratory features suggested this was actually distinct from Kawasaki disease. One of the most interesting features epidemiologically is that the peak in PIMS-TS occurs about a month after the peak in active acute COVID-19 cases. So that was reported in London. Similar has been reported in France and the US. This very much looks like a post-infectious phenomenon. Now, interestingly, just at this point, the adults might have been getting a bit smug, but actually, towards the end of last year, a case series of adults with this condition was reported, was called MIS-A, and identical phenotype to those we were seeing in children, relatively young adults with features of coronary artery dilatation and unfortunately a couple of deaths. Now, during the wave in Victoria in the middle of last year, we were sufficiently concerned that we might start seeing some cases of PIMS-TS that we, a group of us put out an alert through Safer Care Victoria and with help from PEDS and the college, and that was picked up just to make sure that cases weren't being missed. Now, we've only seen about half a dozen cases that we're sure about in Australia so far, which reflects obviously our low levels relatively of acute COVID-19. So there are a number of case definitions floating around this condition. I won't go through them in any detail. They all are roughly the same. And we, or PEDS, a group of us put together one locally, which draws on many of those aspects. So it's really some features of Kawasaki disease, but what is marked is the abdominal pain. These children have rash and abdominal pain and shock. And if you take nothing else away from this talk, that's what you have to remember if you see children in an ED or in a surgery. These are the children who might have PIMS-TS. They're very inflamed. They have very marked cardiac enzymes and a coagulopathy. Often they do not have a history of SARS-CoV-2 infection. They'll be swab negative and no history of an acute respiratory infection, but they're usually positive on serology. There is a WhatsApp group that we've convened through the ANSPID network, the Pediatric ID network, where we can discuss urgent clinical cases, because none of us have any real clinical experience of note with these cases. We're quite well connected with people overseas that we call on. And PEDS is now obviously surveilling not only for Kawasaki disease, but also for PIMS-TS. So I've touched on many of the key clinical features already. This is a disease of older children. There's a male predominance, as there is with many infections and with Kawasaki disease. The abdominal pain is striking. Most of these children are shocked, and they do develop coronary artery dilatation and aneurysms, as you do in Kawasaki disease, but only about a third have had a history of respiratory symptoms. And as I mentioned earlier, with the admitted case series, there are anecdotally some much milder cases in the UK. So I think this is a spectrum of severity. Lymphopenia, as with acute COVID, is a striking feature with neutrophilia and relative thrombocytopenia, raised cardiac enzymes, and lots of inflammation. And I won't go into treatment, but treat it in the same way as we treat Kawasaki disease, i.e. we treat it as a disease that we don't fully understand, but we know the immune system is dysregulated. So we treat it with IVIG, with steroids. Whether we give those sequentially or at the same time is up for debate. There have been ECMO used in a few cases and a number of deaths, unfortunately, but mainly at the start of the pandemic, possibly reflecting that we weren't familiar with this condition. And this paper lined up all the conditions I've been talking about, and I just highlight again some of the features that should make you think of PIMS-TS, but it's a useful summary of the difference between these conditions. And the cardiac manifestations, you do get aneurysms in this condition. They seem to be milder and less severe and resolve more readily than Kawasaki disease, but we do see them, and this is just a case report of a 16-year-old having an MI as part of the PIMS-TS. As I said, I'm not going to go into treatment. I just want to spend the last few minutes talking a little bit about the immunology. I'm not an immunologist, but I think it's really fascinating, and there's some great work out there. All the studies have been limited by the small number of cases. This is a rare condition. This is probably 1 in 5,000 children exposed will develop this at most. So the numbers are small, but the immunology is interesting, and it's characterized, as I say, by hyperinflammation. The T cell subsets discriminate it from Kawasaki disease, and there's some autoantibodies that I'll come back to in a second. And if you just do a PCA plot or of the adult COVID, MIS-C or PIMS-TS and Kawasaki, you can see they look quite different. And there are proteins that, about 20 proteins define that difference. Interestingly, I know this, one list that many don't know, but as part of the infective process, SARS-CoV-2 infected cells produce filopodia that spread the virus from cell to cell. And the production of these filopodia involves casein kinases, these enzymes, and MIS-C or PIMS-TS is characterized by this autoantibodies against this enzyme. So fascinating. I've no idea what it means, but a fascinating finding. The antibody levels against SARS-CoV-2 appear to be somewhat higher in some studies of MIS-C compared to severe COVID. Other studies, again, very small, have found no difference. But what is fascinating in this study, and I might ask Katherine if she's got, or someone else if they got any idea why, is that when you look at those with MIS-C compared to those who are healthy or have Kawasaki or have just SARS-CoV-2 infection, MIS-C patients have no circulating IgG against the circulating normal run-of-the-mill coronaviruses, which is a really fascinating finding. And I don't know what it means, but it's very striking. So finally, as I said very early, that toxic shock syndrome is caused by a superantigen, and a superantigen bypasses the normal specificity of the APC T cell interaction. Now, is MIS-C caused by a superantigen? There is a report came out last week showing that there's a superantigen like motif within the S protein, the spike protein, or near the spike protein of this virus. And possibly this may be a viral superantigen mediated disease, although it's hard to imagine what that superantigen is doing for the 5 weeks between the acute infection and the MIS-C. So just to summarize, these are just a summary of the clinical and some of the immunological features of this condition, a very nice review that came out very recently. We still are at the point of sort of observational immunology. We really don't understand this condition at all well, but it's very interesting, and where there is high transmission of SARS-CoV-2, it's very important that we're aware of it. So I'll stop there. I'd like to thank a very long list of people. I'm sure that's not the complete list. And I think now there is a panel discussion. Thank you.
Thanks, Dave. That was brilliant. Tour de force. Lots of questions for you. So we might come back to some of those in a minute. So Michael, we might go back to you if that's okay. There was a question for you, if you could please comment on the number of cases of public versus private in long-term care facilities by John Litt. Was that something that was an issue, public versus private?
It's certainly a political potential can of worms. So if you mean public as in Victorian funded or supported residential care facilities, then there were, I think, 15 or so cases. If you want to compare that to the others, that is private, whether it be the not-for-profit sector, the charitable sector, or the private sector, they had about 5,000 cases.
Right.
The ratio is about 1 per public facility and 8 on average per private facility. However, you've got to take that with a grain of salt, because as you'll recall, and apart from the issues that they're better funded, better staffed, better supported, better resourced, direct input from hospital facilities, including IPC. The other issue is that many of them are actually in rural Victoria. So given that disproportionately the balance is rural versus metropolitan, they were never going to be at risk anyway. Nevertheless, if you were choosing, that might factor your choice. But the other things, if you look at the Aged Care Safety and Quality Commission feedback from residents, and we've now got tens of thousands of direct feedbacks from consumers, they are telling us that they like smaller facilities and they prefer the public or the not-for-profit.
Okay, thank you. That's really helpful. I just wanted to ask you, you highlighted a number of sort of key learnings or issues from what's occurred, from workforce issues, leadership, logistics, and then you talked at length about the GP response as well. What have we learnt moving forward in terms of, if we have another wave, what would be the biggest issue, do you think, in terms of an effective response? What would you do now? Because you talked a lot about the sort of difficulty in communication and harmonisation.
So we haven't fixed the substrate. And at the end of the day, we're not going to fix the substrate, I imagine, until we actually get the Royal Commission report, which is released on the 26th of March. The government has been holding off. There's been some temporary funding. The length of that funding is unclear. But fundamentally, unless we improve staffing, or until we improve staffing, until we improve training, then we're really going to be challenged. There's no doubt that all of the jurisdictions, Victoria included, and Victoria's been a really good bad example, or an unfortunate example, which has spread across the country, and everybody is setting up their equivalent of the Victorian Aged Care Response Centre and putting things into place. However, it's pretty obvious that there are some significant fundamental issues, particularly in regional and remote areas. Regional and remote simply do not have staffing, or getting staffing there is hugely problematic. And if you wanted to fly staffing into a remote area, where would they stay if you suddenly have a whole lot of your traditional staff suddenly become ill or infected? So there are hugely practical problems that we don't actually have a very easy solution. Metropolitan areas are relatively simpler, but what you're really going to want is that when things fall over, they fall over in a jurisdiction by jurisdiction, not all at the same time, because we needed to borrow or would not have survived from Queensland, South Australia, Western Australia, etc. if suddenly everybody was keeping their own, we would be in a large amount of trouble.
Yeah.
There is a lot of work going on, but there's a lot of work to be done.
Yeah, and obviously training of the workforce around use of PPE and supply is obviously critical as well.
We're all competing for the same workforce, and the problem is, and I'm sure you'll find this in public health, we're on code red almost every day.
Yeah.
Inadequate nurses. Now, once we start pulling nurses away for vaccination, etc., or swabbing, then public hospitals are going to be in real trouble, and then you're going to get direct competition for the same workforce. So we can't instantly increase our workforce. So there are some fundamental issues, and this is not new.
No, and as you say, the Royal Commission will be very helpful. So I might go to Katherine now. There's some great questions for you. Katherine, I was very surprised that in your mild and moderate group, you said 98% of the convalescent group had an IgG response. So I can only assume that's higher in the severe group, of which you had those 5 ICU patients. But there was a question from Rod Pierce saying, what can I tell a patient regarding COVID infection and vaccine about how well protected they are? In other words, if they've had infection, how well protected are they? And I assume post-vaccination as well. What would you, and you also commented about the antibody avidity and the quality of the response. So can you make any comments around that?
Yeah, so obviously traditionally assessing antibody levels, and especially the neutralizing antibodies, has been associated with protection. However, we still do not know about the durability of antibodies after SARS-CoV-2 infection, and us and other groups obviously are studying it, as well as the levels. What are the levels needed for protection? But what we know that the majority of the patients do seroconvert. They have some level of antibodies for neutralizing or non-neutralizing function, but also antigen-specific B cells and T cells. So in patients, there is some level of immunity that memory immune cells that after subsequent infection will lead most likely to a rapid response.
Yeah. And you talked as well about the immune perturbations in the severe group. What learnings do you think we've got from that so far in terms of moving forward in managing those patients? You talked about the IL-6 response. What were some of the other learnings from that work?
Yeah, so it was IL-6, IL-18, and the soluble IL-6 receptor that were markedly increased in the ICU patients. So obviously now we know what are the potential biomarkers for predicting which patients will have severe disease and may end up in ICU rather than have a more rapid recovery. And apart from those cytokines, looking at the activation level and the breadth of immune cells is also important to understand the potential severe disease.
And can you make a comment about the antibody response in people who experience mild infection in the community? What proportion of them would you expect to have an IgG response, and how does that relate to the need for vaccination?
So in our cohort, all the patients with the exception of one seroconverted, people who were the COVID patients from the community had antibody responses, but usually, as published by others as well, more severe disease leads to higher levels of antibodies. It's still with respect to the vaccination.
It's a question currently being asked a lot at the moment.
I know, as an immunologist, I'd say that vaccination after having SARS-CoV-2 infection would be beneficial and would further boost those memory B cells, memory T cells, and the levels of antibodies and their quality.
Yeah. Dave, before we get into some of the PIMS-TS questions, what would be your comment about the antibody response in children post SARS-CoV-2 infection compared to that 98% immune response in adults?
Yeah, I think it's something it's something you've been part of those discussions we've really struggled with, haven't we, in terms of trying to just find some serological marker of exposure for our studies, because we can't use the symptomatology given it's often asymptomatic, and it looks like I don't know. I mean, it's it's impossible to keep up with the literature, but it was looking like the serology, if positive, was waning reasonably quickly. Perhaps there was a lot of hope about salivary antibodies. I guess in terms of, I've approached this largely from a research, finding children who've been exposed to identify those who've been mildly affected. So as I think the jury's still out, I'm hoping that, in this fast moving field, we'll see some sort of biomarker of identifying these children. But clearly in that family, the Discorvid family, that was a salutary lesson that you can be infected with really, if you rely on your traditional tests, you're going to miss these children. And there may well be a lot of children like that out there. I mean, others, maybe yourself or Katherine may be better placed to comment.
Yeah, I mean, as you say, in that Discorvid family, fascinating that the salivary IgA was positive in the kids, but of course only one seroconverted and they were all PCR negative. I'm going to ping the paediatric vaccine questions to you, Dave, if that's all right, or maybe Katherine wants to pipe in as well. So we've got two, one from Rod Pierce has asked, vaccination in children is planned to happen last. Is that still appropriate? And Sonia Elliott has also said, shouldn't children with significant comorbidities be a priority group for COVID vaccine? So some thoughts around COVID vaccination in children?
Yeah, so I'm batting a long way out of my crease at the moment. But given that Joe Root made over 200 again, I feel slightly confident. Yeah, I mean, I think it is probably appropriate on what we know so far. It does look as though this is a mild, not always, but usually a mild disease or an asymptomatic disease in children. And it does make sense to protect the most vulnerable. It doesn't look as though schools, I don't want to get into the schools debate, but
Yeah.
as paediatricians and parents, we are very keen on keeping schools open as much as possible. We don't know, actually, as far as I'm aware, about the comorbidities that are clearly increasing risk in adults and children. There was some discussion in the PIMS TS literature around children with obesity possibly being at higher risk of more severe disease, but I don't think we've seen we've seen lots of case reports from children with comorbidities, but I don't think we've seen a definite signal from a particular group who are at higher risk. And there are obviously plenty of groups that you might consider would be at higher risk, but I don't think that's at all resolved. So I think that it's a good question for Sonia, and I think it's probably an evolving, we just have to be keep aware of, if there is a signal, then we change the priority.
Lots of questions about COVID vaccination and immunity. So Katherine, back to you. Can you comment on the interval between COVID infection, whether it's mild or moderate, and offering vaccination?
No, I don't think such studies been done.
I think currently, Dave might like to pipe in here, I think WHO and others are saying 6 months, but then there's discussion of whether it might be shorter than that. I don't know if you've read anything else.
No, I think I don't have anything to add to that. I think you're probably across the vaccine policy area better than me.
Our audience are asking lots of vaccine questions, so I'm sorry to be pinging these at you.
Well, it's that kind of conflict.
Yeah, absolutely. All right. So Dave, I just wanted to ask you in terms of PIMS TS, it's absolutely fascinating. And the comparison you gave across KD and toxic shock was fantastic. Firstly, can you comment on the drop of KD cases during COVID? I was interested why you thought that might have occurred, even though it was maybe only mild compared to looking at that PEDS data.
Yeah, I'm not that convinced by it. I mean, it's probably true. I mean, I think PEDS do a very good job, so I don't think that's the source of the issue. There is some variation year on year, and there's obviously some seasonality around KD. We're just starting to see some reports possibly from high incidence countries, which may be more relevant. I mean, it's hard to believe that even in a situation where parents may be reluctant to bring their child to hospital, or there may be difficulty with access to healthcare. If you've got a 1-year-old with a fever of 39 to 40 for 5 to 7 days, you won't bring that child to hospital. That child, I think, will find their way to hospital. If it's true, then there is, my personal view, without going into details, is that Kawasaki is caused by one or more infectious triggers. I suspect more than one. One of which is a superantigen, one's possibly a virus, and I suspect there's obviously much less opportunity for those triggers to circulate in the general community, and then to pick off the children who have a genetic predisposition. So I suspect that's what's happening, rather than a problem with access to healthcare. So I think it's probably true. The question is the extent and what it might tell us about the cause of KD.
And then talking about PIMS-TS, which I must say I prefer that term rather than the other one, you said we'd only seen about half a dozen, so 6 cases or so. There was a huge amount written about it in the media, though, when it sort of blew up here. Can you comment a little bit more on the treatment separate to just the IVIG and steroids? There's been a lot of sort of other treatments floated. You mentioned briefly something about autoantibodies or maybe anticoagulation therapy.
Yeah, so 4 confirmed cases and a few that were are still up for debate, didn't quite fulfill criteria. So in terms of treatment, and we're very fortunate in the pediatric ID world that we can really phone a friend, and Mike Levine, who's another giant of pediatric ID, was on the end of a Zoom call within about half an hour of our first case in Melbourne, and that was invaluable because they'd had a lot of experience. So the mainstay is IVIG. Most of these children do respond to immunoglobulin, as Kawasaki disease is that similar large dose. You have to be careful because they've got myocarditis. You're giving them a big oncotic load. You can push them into heart failure, but that's the treatment. There was a study came out, I think, last week showing that perhaps giving steroids at the same time in the more severe cases does maybe slightly better than giving it sequentially. That's still out for debate. There has been talk about venous thrombosis in these children. They've seen a few cases in the UK, so some anticoagulation is recommended. Low dose aspirin, antithrombotic effect to protect the coronary arteries, but it's all very non-specific. And the data on biologics as almost the third line is really not that convincing. And I think it depends which specialty you ask, whether you go for anti-IL-6, anti-TNF, anti-IL-1. I think probably anti-IL-1 makes most sense. Most of the inflammatory response seems to come from monocytes, but I think it's down to sort of it's anecdote-based medicine, really. So it's non-specific treatment because we don't really understand this condition. So
yeah,
yeah.
And there's just, I think I might sneak in one more question if I'm allowed, and then I have to wrap up. There was a question here by Rod Pierce about long COVID disease, and is there such a condition, and is that an issue with children?
Look, I think it is a condition. I only read what others read, and it's to date it's largely been in the press rather than in the scientific journals, but it does very much seem to be a condition, particularly in women. And a number of reports. I think immunologically and sort of using the full arsenal of omic technologies to look at this is going to be really fascinating. And I think it's important that we look in children. We certainly see in other infections that even mild infections can increase your cardiovascular and metabolic risk. And whether that's true of SARS-CoV-2 is important. So I think this is something that is an unfolding story. And I think it's really important that we don't forget the children in that story.
Yeah, absolutely. Look, there's so many terrific questions coming through. We're going to have to wrap it up there, I'm afraid, but I really would like to sincerely thank Michael and Katherine and David. Thank you very much, everyone, for a fantastic session.